I am Constantino Macias, one of the organizers of this symposium, and Helen. Oh, Helen is the other organizer, and she likes to sit in the relative back. So thank you very much for coming and contributing. It is already 31 years since Ryan and Rand alerted us to the fact that signals used in made choice context may owe their shall I say, configuration, to avoid the word design, to pre-existing perceptual predispositions on the sensory system of intended receivers. This proposition made sense, and hence it spawned a plethora of studies that convincingly demonstrated that indeed responsiveness to key features of mating signals often precede the evolution of those signals. Spearheaded by Alex Basolo's work on conspicuous visual ornaments in sortal fish and Mike Ryan's increasingly sophisticated analysis of Tungara frog acoustic responsiveness, we came to accept the fact that sensory biases, particularly in the acoustic, visual, and mechanical modalities, frequently underlie the evolution of ornaments. Yet mate choice or mate assessment is only one context in which organisms use environmental cues or signals to orient their behavior, and hence sexual selection only one of many biological processes where we would expect sensory biases to shape biological interactions. Indeed, any goal-oriented behavior uses some type of sensory input to which natural selection tunes the sensory system so that efficient responsiveness to biologically relevant cues is ensured. Sensory biases then arise either through this process or because of fundamental physiological constraints and are open for grabs, leading to sensory traps and sensory exploitation. In concocting this symposium, we speculated that perhaps then sensory biases are ubiquitous. They should have appeared frequently in the history of life and should involve all perceptual modalities. We also wonder whether they have consequences, not only in mate choice, but in other biological contexts. In the following talks, we shall get directions towards answering our suppositions and surely gather new insights to inform our study of sensory bias biology. Thank you very much for coming to listen and to contribute to this symposium. And I leave you now with Becky Fuller from the University of Illinois for what will be a very interesting opening talk. Thank you.
Thank you very much. I want to start by thanking Helen and Constantino for organizing this symposium and for inviting me to be here today. And I just want to say I'm really excited to hear some of these other talks by this exciting lineup. So, yeah, just to start off from a really general standpoint, I think we all know that in many species, males evolve costly secondary sex traits. So here in this figure here, we have this brightly colored finch. We have these brightly colored guppies and bluefin killifish. We have this sea lamprey who's presumably stinky, or I should say smelly in a good way. And we have this male Tungara frog who's calling his guts out. So, yeah, and so one of the questions that we often address, and a classic question that we teach our undergrads is, why should males evolve traits like these? And at one level, we have a really cheap answer, and it's that males evolve traits like these because it increases their male mating success, either through the effects of competition among males or by currying the favor of females to their female mating preferences. And indeed, just going to go forward, we know that in many species that male secondary sex traits evolve due to the effects of female mating preferences. Now, I'm going to argue that in the area of sensory bias that we've predominantly asked three questions. The first question is, why do male secondary sex traits take the form that they do? Why do guppies have orange spots? Why do lampreys have certain smells that they have? And at one level, I'm going to argue that we, again, have a sort of cheap answer here, and that the answer is that males evolve traits that meet the mating preferences of females, provided the costs and benefits of doing so and any limitations that's posed by the genetic constraints that might be there. Now, the more challenging question is trying to understand, why do females have the preferences that they do? Why do females behave in the way that they do? And I come from the University of Illinois, and we have a large, strong group working on animal behavior, and my colleagues there often remind me that we need to be thinking about behavior from multiple vantage points. We need to be thinking about them through the lens of the great Dutch ethologist Nico Tinbergen's 4 questions. And so I'm going to argue that we actually have been doing this. In the field of sensory bias and sensory drive, we've been doing this for over 20 years. And this is what I want to talk about today before I go to talk about my research. And I'm also going to argue that this is a slightly broader question that informs our understanding of what accounts for the evolution of female mating preferences that, in terms of sensory bias, might actually be a smaller subset of Nico Tinbergen's 4 whys. And I want to start out by acknowledging the great pioneers in this area: John Endler, Alex Basolo, Mike Ryan, and Mary Jane West Eberhard. Okay, so let's do it. So what I want to do is I want to go through Nico Tinbergen's 4 questions and their application to sensory bias. And the first sort of vantage point that I want to talk about is phylogeny. It's the history of the changes of the behavior or the trait across many generations. And I think it's clear that the earliest evidence for sensory bias came from really clear phylogenetic patterns that we see regarding the evolution of traits and the evolution of preferences. So Alex Basolo, this is classic work now, where Alex Basolo went and mapped across species whether or not males have this, this swordtail, this elongation of the caudal fin, and then she was able to map it and show when it arose. And then, using very clever experimentation, she could go and ask, even in species that lacked the sword, whether or not the females had the preference for the trait. And lo and behold, the preference evolved long before the trait did. Now, this is a case where we map behavior and we map the trait, the male trait, onto the phylogeny. But we've also done things where we have mapped sensory system properties and male traits onto phylogenies as well. So an early example of this came from Andre Fernandez and Molly Morris, who mapped across primates whether or not the primates had dichromatic or trichromatic vision. And then they also mapped the lineages in which there was red skin color and red pelage color. And what they were able to show was that, lo and behold, trichromatic vision evolved long before we ever saw any of these reddish colorations in these primates. And again, we would take this as evidence for sensory bias. Okay, so that's the phylogeny. Let's talk about mechanism, the mechanistic explanations for behavior. And again, I would argue that we have been doing this for quite some time in the area of sensory bias and sensory drive, and that we've been really interested in the evolution of the properties of sensory and neural systems. So just early work by Mike Ryan, one of the leaders in this field, looked at the auditory tuning of the Tangara frog, the auditory system, and saw the parts of the sound space where frogs are most sensitive. It's where that curve is lowest. And then he compared this to the frog call properties and showed, lo and behold, that if you look at the area where the frogs are most sensitive, it roughly corresponds to the area where males are putting their most energy, their biggest bang for their buck, into these call properties. Okay, we have been, in the area of vision and visual ecology, we have been long fascinated with the various details of how animals see, right? And there was actually there were great talks yesterday. There were some ones on frog vision, the size of the eye, the shape of the lens. People worry about flicker fusion. How many cones do we have? Which cones do they compare? Just sort of figuring out all these details about how animals go about seeing the things that they do. And we sort of start at the level of comparing across species. So we're humans, and we have 3 cone cells. We have a blue and a green and a red, and a raw that is indicated, the sensitivity by that dashed line. And yes, my name is Becky, not Bucky Fuller. Sorry, it's my one joke. Okay. Yeah, I visited at the University of Wisconsin. Sorry, I think it's funny, but everybody else thinks it's super funny. Okay, this is in comparison to the bluefin killifish that we study in my lab, where these animals have 5 distinct cone cells. And people always say, oh, birds and insects have 4, they're so complex. Yeah, sorry. These fish and many fish actually have 5 cone cells. They have a UV, a violet, a blue, a green, and a red. Now, this stands in contrast to their major predator, the largemouth bass, that only has 2 cone cells, and for all intents and purposes is sort of blind in this blue and UV range. Now, the other things that we have figured out is that not only are there differences across species, but we also know that populations can vary in their sensory system properties. And I'm going to show my age here and tell you that when I started as a young graduate student, there was this sort of idea out there that these sensory systems are so complex, they're really constrained, and it's almost like we thought of the sensory system as like a big house of cards. It's like a big house of cards, and if we pull one of those cards out, the whole thing is going to fall apart. But that sort of conception of the complexity of the sensory system is wrong, and that, as soon as we started scratching below the surface, we quickly found population variation in sensory system properties. So these bluefin killifish that I work on can occur in different habitat types. They occur in springs that are crystal clear with tons of UV and blue light coming through, but they also occur in these swamps, and the swamps are more tannin stained, as you can see in this picture below. And if you simply go and compare all the populations have all 5 cone cell types, but they differ in the relative abundance of those cone cell types. So spring environments that are shifted towards lots of UV and blue light these animals have higher relative amounts of UV and violet cone cells, and the ones that are in the swamp have higher relative amounts of yellow and red. And there's lots of evidence for population variation in sensory system properties. Jenny Bauman just talked yesterday about these sensory system differences across these stickleback populations in Iceland. Yeah, and I'll also add that population variation is really prevalent in aquatic systems because our lighting environments are so very, they differ a lot. Okay, so we talked about phylogeny, we talked about mechanism. Let's talk about ontogeny, the developmental explanation for changes in behavior, and I'll add changes in sensory system. So here we can look at the effects of development and plasticity on sensory system preferences. And to return to this bluefin killifish system that we've been working on, we can go, they're external fertilizers, so we can make a big clutch. We can take half the eggs and put half of them in clear and half of them in tea-stained water. We can raise the babies up and look at the expression of various things in the eyes. And we're actually finally starting to do some transcriptomics, so stay tuned for that. But our early work showed that when you do this, when you raise animals in clear water and tea-stained water, that lo and behold, you get really large shifts where you get higher expression of the SWS1 and the 2B, which corresponds to the UV and violet cone cells in the clear water conditions, and you get large shifts towards the longer wavelengths in the tea-stained conditions, where you get higher expression of the LWS and the RH2, which correspond to the green and the red cone. And there have been oodles and oodles of studies since then showing that there are plastic visual systems and associated behavior. And if you think I'm talking fast, just wait. So, okay, carotenoid intake affects opsin expression in guppies, and it also affects oil droplet properties in birds, which filters the light. We know that depth in the water column affects the filters used by mantis shrimp. You can take the little larvae and put them into shallow water or the really deep water, and they will get completely different filtering properties to their eyes. We know that birds living near airports shift the change in the timing and the properties of their calls. We know in cichlids and actually tons of fish that lighting environment and thyroid hormone alter opsin expression in cichlids. And going more towards behavior, so in this swordtail carasin, these males have this little filament and this little thing that sort of hangs out, and people have proposed it's this lure, and that it looks like an ant, because these guys go and eat stuff off the surface. When they raise female swordtail carasins and they feed them ants, they're more likely to be attracted to this lure than if they're fed other things. Okay, so we've done 3 of Tinbergen's 4 questions. Let's do the 4th one, the functional one. For Tinbergen, it's a behavior that solves a reproductive or survival problem in the current environment. What's our predictions for sensory bias? So, let's talk about this for a second. So, I think that we can all agree that sensory systems should encode external stimuli into internal biological signals in an efficient manner. Nervous systems are really costly, and so you want sensory systems that get the relevant stimuli into the body in an efficient manner. I think we can all agree that animals need to find food, avoid predators, find proper habitat, and that this creates natural selection on sensory systems to get those external stimuli that are relevant so that animals can use that information. And then I think we can agree that females need to find mates and that selection should maximize the benefit to cost ratio of mating, given the costs and benefits and, again, whatever genetic constraints there might be. And this is sort of where I think the rubber hits the road. So our sense, in terms of this functional explanation from Tinbergen, I think the way that this works is that we have natural selection on non-mating behaviors, on sensory system properties. And the question is, to what extent does this create a correlated response in mating preferences? So to what extent are these two correlated? And there's clear evidence from Bossolo and others that we can have preferences that evolve in one circumstance and then they get used in another. The real question here is, to what extent can selection, if these two things are in conflict, to what extent can it pull these things apart? So to what extent can selection act independently on each behavior? And to what extent does sharing a common sensory system constrain the evolution of behavior? Okay, so for my remaining time, I want to tell you a little bit about work that we've done in bluefin killifish to try to answer some of these questions. Sorry, I forgot. I'll add that the environment under which all of this takes place can also have an effect in shifting sensory system properties based on whether, in the case of bluefin killifish, whether it's a clear water condition or a tea stain condition. And that sort of variation there, we can use it to sort of test the sensory bias, because as we get differences in sensory system properties between springs and swamps, we can also look for correlated effects in mating behaviors and mating preferences. Okay, so let me tell you a little bit more about bluefin killifish. So bluefin killifish they're beautiful fish. Yeah. I'm getting more people to study them, but so it's slowly but surely. Anyway, so they're beautiful fish, and the milk coloration is really variable. So males can have a solid blue anal fin like this guy shown on top. It can have a solid red anal fin, a solid yellow anal fin. There can be combinations of blue and red and combinations of blue and yellow. And we get multiple morphs in all of the populations that we study. Now, the other interesting thing that I already mentioned is that these guys live in different lighting environments. So sometimes they're in clear water conditions, sometimes they're in tea stain conditions. And we can see the difference between these populations with our eyes, right? But if you go and take your spectrophotometer and you point it up and you say, what light am I getting? You will see that the spring lighting spectrum is this really broad spectrum with tons of UV and blue, and the swamp spectrum is depauperate in the UV and blue wavelengths. And so, one thing we did many, many years ago is we went and did a large statewide census. We collected as many animals as we could. We measured the lighting properties, and then we just simply looked at the relative abundance of these various color morphs. And what we found that let me go back. Each one of these dots is a different population in Florida, and so it's showing you the relative abundance, or the proportion of males that had solid blue anal fins. And so we get more males with solid blue anal fins in swamp-like conditions, and we get fewer of them in spring-like conditions. And I already showed you earlier that we know that the sensory system properties vary between swamps and springs. So we know that both the male ornamentation and the visual system properties vary with the lighting environment. Okay, so the question is, are there strong correlations between sensory system properties, foraging preferences, and mating preferences? What are the effects of genetics and environment on these traits? Okay, so I'm going to tell you about an experiment that we've done where we've tried to tease apart the effects of the lighting environment on foraging preferences and mating preferences. And I want to make the point that these differences in the lighting environment can have effects in 3 different ways. The first off, you can have genetic differentiation among populations just due to the fact that we have populations in springs and populations in swamps, and if you get natural selection and sexual selection operating under these different environmental conditions, that we can of course get genetic differentiation among populations. We can also have effects due to the rearing environment. So simply being reared in a clear water condition or a tea stain condition can and does have long-lasting effect on phenotypes. So when we can raise babies in clear water and tea stain water to mimic the effects of springs and swamps. And then the testing environment can immediately affect the perception of the signal. And when we think about sensory drive, I think that this is the main effect that we think of in terms of the effect of the environment. So, if we have animals in a tea-stained environment versus, a clear water condition, this is going to affect the distribution and the abundance of the various wavelengths at depth. It's going to affect the background that colors are perceived on, and it's going to affect the transmission between the object and the viewer shown here. Okay, so we did an experiment to try to get at this. So we did an experiment where we did crosses between spring males and spring females, swamp males and swamp females. And then we took the offspring and we raised half of them in clear water conditions and half of them in tea-stained conditions to adulthood. And then we tested these guys. We tested them in clear water conditions and in tea-stained conditions. And then we measured the behavior there. So we could look at the effects of genetics, the effect of rearing environment, and the effect of testing environment. I'll also add that we used certain male color patterns for both of the two populations, but I'm not going to be talking too much about that here today. Okay, so let's talk about the foraging preferences. So our color preference assay is, as always, inspired by the great Helen Rodd. So Helen, as I think many of us know, painted these Trinidadian dimes and chucked them in different populations and saw how animals pecked at these. And so we did something very similar. We took a Petri dish, we put different colors on this Petri dish, and then we simply let it float down and let animals peck at that to infer either I don't know if foraging preference is the right word, but color preference in a non-mating context is definitely a correct way to view this. And of course, we did this under both clear water and tea-stained conditions. So I'm going to tell you about the relative effects of genetics, rearing environment, and testing environment. But I'm going to tell you right now, for color preference assay, there were no effects of genetics. There was no differences between our spring and swamp populations, and there was no evidence of among family variation. Okay, but what we did see is a really interesting pattern where we got our highest level of preference for pecking at blue when animals are reared under tea-stained conditions and are tested under tea-stained conditions. So this is we can see this here. We have the rearing environment, whether it's clear or tea on the X, and the testing environment in the white and black bars. So this suggests that there's something to having this color preference for blue under tea-stained conditions. Now, in this study, the pecks at blue were actually relatively low in comparison to pecks for red. But we found a similar result in another study. So in another study, we went and we took animals from a confluence between springs and swamps, and we did a similar type of thing where we tested their pecking preference in clear and tea-stained water, and we did it in the morning and at midday and at dusk. And we could see here that they never peck at the blue spots when it's clear. They only peck when it's tea-stained conditions, and we get an effect of time of day, where they peck more at midday. Okay, in terms of the choice assay, so here we did a one-choice assay, which is, so we left, let me go back. Bluefin killifish, one of the great things about them is they will spawn every day for a couple of weeks, and then they'll take some time off, and then they'll go spawn some more. And so we took advantage of this fact, and we put a female with a male in a particular lighting environment, let him mate, gave her a day off, put her with another male at 8 a.m., pull her out at noon, see how many eggs she lays. And we let the female mate with these different color morphs under these different lighting environments. And we can infer the preferences from the number of eggs spawned with each male. And what we found here is we found an interaction between genetics, rearing environment, and testing environment. And so we only found a strong preference or a preference that deviated from the null expectation for blue males when females had swamp parents, when they were tested under the tea-stained environment, and when they were reared under the tea-stained conditions. So, where are we here? So, the thing I find that's interesting is that there's different variance components that contribute to these foraging preferences and to these mating preferences. And both of these favor blue under tannin-stained water. So for our foraging preferences, we see that it's an interaction between testing environment and rearing environment where we get these blue preferences. For the mating preferences, you have to have the right genetics, and then you have to be, again, in the right rearing environment and the right testing environment. But both of them favor blue in tannin-stained water. So at one level we have a correlation, and at another level we don't, right? Yeah. Okay, so this leads to our sort of interpretation, which is that we're becoming increasingly convinced that lighting environments have tons of effects on lots of different traits. But these traits aren't necessarily correlated with each other. Yeah, but foraging and preferences and mating preferences appear to be independent of one another. So to take us back, I hope that I sort of have convinced you that we use the term sensory bias in lots of different ways. So we sometimes refer to it as a phylogenetic pattern. We sometimes refer to it, when we're talking about the various sensory properties of the animals. Sometimes we talk about it as to the differences that are induced when they're reared under different conditions. And sometimes we talk about it in this more sort of quantitative trade-offs, direction of selection framework. And I want to add each of these touches on a fascinating aspect of biology. Comparative sensory ecology, I can take that stuff all day. Rearing these animals under different lighting habitats and seeing the effects on the visual system, I think that's great. But the sensory bias model, as a model for the evolution of female mating preferences, involves natural selection on sensory systems and non-mating behaviors and correlated responses on preference. And so whether or not these, it's undoubtedly, the selection in the past environment will affect the preferences that the animals first have. But whether or not these actually constrain the evolution of adaptive choice and whether these females are exploited by their sensory systems is unclear. I think there's tons of intriguing questions that remain. I want to give a shout out to all the greats in this field, including the great Helen Rodd. With that, I'd like to thank the University of Illinois, National Science Foundation, my lab, my collaborators, and you all for being here. So thank you very much. No, go. You have time for, or do we need to move on, or are we good? Question?
No, that was really great, thanks.
No.
One of the features of many sensory systems is adaptation, so those environments efficiently, producing phenomena like instances. Is that something you've looked at in killifish as, having evolved in some of these cases in different light environments?
I can sort of respond to that sort of yes and sort of no. So the idea of color constancy, right, is that, if you go to these different habitats, right, your visual system should adapt to try to keep things relatively constant. And in fact, in the Opson expression stuff, you can predict either pattern, right? So if you're trying to create color constancy, then you would increase your UV expression under T-stain conditions to bump up your sensitivity on that to make up for the lack of the wavelengths there. But if you're, there's a matching pattern and a compensating pattern. I think the thing that's hard to do is, there's only so much that you can correct for color constancy when there's no wavelengths, right? Or when there's practically, no wavelengths at depth, right? So we've thought about it to the extent that I just, said. That helps. Question?
Is there any learning on this?
Is there any learning? Do they learn? I'm trying to think.
They have something.
So Ben Sandkham was an undergrad in my lab. I was like, Ben, did we do learning? We haven't looked into learning. So I don't know the answer. Like, are you asking whether they can learn some things better than others? Like there's certain colors that are salient, and so they could learn a blue easier than a red or whatnot.
If there's, for example, some imprinting on if certain individuals that have grown in environments in which certain color is more common, they have certain eyes for that color.
We haven't looked at that, but that might be an interesting thing. So, okay.
So thank you very much.
Hi, good morning, everyone. Thanks so much for joining my talk. Today, I'd like to talk about the evolutionary relationship between visual preference and the plumage white dot patterns in Ecuador finches. The sensory bias hypothesis predicts attention or preference in non-sexual context, such as foraging and predator avoidance, can influence the evolution of signal trees. The yellow band in this species tails is one of the examples of the sensory bias hypothesis. In birds, the sensory bias hypothesis has not been well examined, but birds use various signaling traits, such as dance, song, and color. Many researchers have focused on plumage colors. But how about the plumage patterns? The meaning of pattern here is a combination of color and the shape of pattern motif. So we don't intend to use the color badge or something. Unfortunately, studies of the evolution of plumage patterns is far less well understood. Compared to other patterns, dots are particularly common and known to have signaling function. Here, we tested whether the sensory bias hypothesis can explain the signal evolution of plumage dot patterns using the family Estritidae. The sensory bias hypothesis has mainly explained the evolution of male sexual signal trees, but we predict this hypothesis could also apply to the evolution of signal traits shared by male and females. If both male and females prefer the same food, the preference for finding that food could evolve in male and females. This shared preference might influence the evolution of signal traits in both male and females, leading to these trees resembling their preferred food. So we selected this family as our research subject for two reasons: their plumage patterns, white dots signaling function, and diet. First, their white dot plumage patterns are known to function as sexual or social signals. Over 25 of species have white dots, and both male and females often have similar dots. Male in such species often show a unique courtship display. This photo is one example. This is a male starfinch. They fluff up and fully display only the area existing white dot plumage patterns. Second, estrel finches either two types of pale and gregarious food. All estrel finches forage tiny grain seed, and some species like to eat pale-colored, small and round invertebrates, such as termites. We will refer to this as termite eating hereafter. Interestingly, around 68% of termite eating species have white dot plumage patterns. For these two reasons, we have examined whether foraging related to visual preference contributes to the evolution of the white dot plumage patterns on estrel finches. Specifically, we predict foraging promoted visual preference for white dot-like patterns, and it may be associated with the evolution of plumage dots in living estrel species. Then there may be an evolutionary relationship between diet and white dot plumage patterns. Our previous study have shown white dot plumage patterns could have related to termite eating, and termite eating evolved before the evolution of plumage white dots. Also, we found one estrel species showed a strong visual preference for white dots, abstract white dot patterns. To estimate whether such visual preference for white dots is a common ancestral trait. We investigated this preference using behavioral experiments on several estrus species and compared their responses considering phylogenetic relationship. Here are our two possible predictions. In phylogenetic trees, a visual preference are in blue and white plumage dots in olive green. If the visual preference existed in their common ancestor, living estrus species will possibly share a visual preference for white dots, regardless of whether they have white dot plumage patterns. In addition, if the visual preference was originally for foraging, a stronger visual preference for dots may be shown when they are hungry. Alternatively, when dot preference does not pre-exist and evolve in specific living species, among species variation in diet or plumage dots would probably link this preference. This is a phylogenetic tree of estrus finches and closely related species. So estrus family is here. So we used 95 individuals of 12 species and an average of 8 individuals per species. We included those with and without white dot plumage patterns and termite eaters or not. In the experiments, so we present pair of abstract stimuli consisting of black background with white dots or stripe patterns. Visual preference was measured by the total frequency of gazing at pattern stimuli per 1 hour experimental trial. Here is an example. And this is an experimental cage, and the stimuli were placed here and marked S. If the subject prefer dots over stripes, we can predict the subject has hunger-related or hunger-neutral visual preference for white dots. To test this, we observed subject responses to pattern stimuli under food-deprived and food-supplied conditions. The experiment was conducted over 3 days. First, we took a habituation period in experimental cage. The next day, we conducted the experiment under the food-deprived condition to test hunger-related visual preference. We expected hungry estrus finches would gaze at dots more often than at stripes. Then we did the experiment under the food supply condition to test hunger-neutral visual preference. If estrus finches have this preference, they might still prefer dots even after they have had experience with dots didn't provide any reward. This figure shows the result of experiments under 2 conditions. The x-axis means dots and stripes under 2 conditions, while the y-axis indicates the total frequency of gazing in 1 hour. Each plot shows the mean and the standard error of gazing frequency per species. We found the total frequency of gazing to the dots was positively significant than stripes under the food-deprived condition. Species generally preferred to white dots over stripes. Under the food-supplied condition, gazing was observed, but the difference in responses was only marginally significant. We did not find a negative or positive significant effect of sex of sex, eating termites or not, and the presence of white dot plumage patterns in the responses. This is a result of ancestral reconstruction based on our behavioral experiments. Blue means the probability of visual preference for white dots is present, and yellow is absent. I'd like you to focus on the root of the phylogenetic tree. The ancestor states of hunger-related white dot visual preference were moderately likely to be present in estival common ancestor. However, hunger-neutral visual preference for white dots was 50-50 in the common estival ancestors. This pie chart shows whether the common ancestors had white dot plumage patterns based on our previous studies. Blue indicates the probability that white dot plumage pattern is present, and yellow indicates the absent. Unlike visual preference, common ancestors were less likely to have had white dot plumage patterns. Our findings indicate the common ancestors of male and female estival finches may have had a visual preference for white dots related to foraging. It's possible foraging for grain influenced this preference and the evolution of dot patterns in their plumages. While the preference for white dots over stripes was not statistically significant when food was provided, but the trend still aligns with the idea of the ancestral visual preference for white dots. Here's our take-home message. So the pre-existing visual preference originally shaped for foraging could play a role in the evolution of plumage ornamentation, and birds have many unique ornamental traits, but we don't fully understand why they evolved. So there are still many mysteries about these traits. The sensor bias hypothesis might explain some of these, but more research is needed. So we sincerely appreciate the organizers for giving us the opportunity to be here today. And thank you so much for joining my talk.
Thank you very much. We have time for one question.
Do you think, thanks for the talk, do you think there was any effective learning in terms of the order that you did the hunger experiments?
Sorry, I don't think so, because we use the very abstract, unnatural stimuli. So our subject did not experience such patterns. So we removed the learning experiment to use such stimuli. It's okay. Thank you.
Okay, thank you very much.
Okay. Well, it's really such a pleasure to be here. Thanks everybody for making it to my talk and the opportunity to share some results with you. So I study the sea lamprey. For those of you who aren't familiar with sea lamprey, they're this really fascinating but destructive jawless fish that invaded the Great Lakes over 100 years ago now. And have done a lot of damage to the fisheries there, and that really was kind of my entry point into studying pheromone communication in them, and continues to motivate a lot of the work that I do. But I think the data that I've been collecting, I guess going way back to when I was a graduate student, it all got really interesting to me when I was actually sitting in a class that Jenny Bauman taught on animal behavior, and I learned about sensory biases for the first time. And I've spent kind of a surprising amount of years now kind of following up on that. So I think this is actually the same schematic that Jenny showed that caught my attention, and it's really one of my favorite examples of one type of sensory bias, which is called the sensory trap. And what this illustration is showing, this is a couple of papers that Heather Proctor wrote in the early 90s, is water mites, where they feed on these copepod prey, and those prey produce vibratory cues, and the males essentially mimic those cues with their leg during mating, and when they do that, the females clutch onto them as if they were a prey, and the male then takes the opportunity to mate with her. And when I was in Jenny's class, kind of started to notice some parallels with the data that I was collecting, but what I want to talk about today is mostly not the origin of female preferences, but more so what happens next once females are deceived, essentially, or receivers in general are deceived by these traits that are essentially mimics. But before I get into that, just a bit about lamprey and a little bit about why we think there is a sensory trap underlying how they communicate with pheromones. So lamprey start their lives as larvae in streams. They just burrow in the muck and filter the water for 3 to maybe 17 years. It just could be a long time, often probably an average of 5. And after this time, they move down into the Atlantic Ocean where they're native, or the Great Lakes where they're invasive. And there they parasitize on fish using their nasty jawless or tooth sucker mouth thing. It's pretty gnarly. And after doing that for about 1.5 years, they stop feeding and they prepare for their single spawning season, which they do in streams, and they navigate 2 spawning streams following the odor of those larvae that are living in those streams year-round, and we call that the migratory queue. And this happens in the early spring and kind of as the temperatures start to warm up. And one component of this multi-component migratory queue that they follow into spawning tributaries is 3-ketopetramizanol sulfate, or 3-KPZS. And 3-KPZS is a bile acid that's synthesized in the liver and involved in digestion. And larvae just release it, excrete it through their feces, along with, like I said, some other components that are involved here too. And both males and females during this pre-spawning migration orient upstream, kind of in not a very targeted way, but a general movement upstream into tributaries that have 3-KPZS in the water. After a couple of weeks, as the water temperatures continue to warm up, the males and females become sexually mature and are physiologically ready to spawn, and the males construct spawning nests on gravel bars. And the males signal to females with another multi-component pheromone, which interestingly has the major component, again, of 3-KPZS. So kind of these data, along with some phylogenetic comparisons that I'm not going to talk about today, support the hypothesis that this non-sexual orientation to 3-KPZS was the original context in which females evolved this preference, and then males evolved to exploit that preference by signaling with 3-KPZS during spawning. And just a quick note and I'm not going to get into a lot, but of what happened next here. One thing that we have observed is that females seem to benefit from this. They have actually evolved a stronger response now in the sexual context compared to the non-sexual context. And this makes sense because they only have a short window of time to spawn before they die, so they really, it makes sense that they would benefit from a really efficient way of finding males. However, what I want to talk about today and what I've been thinking a lot about is what this might mean to females as they're navigating through these streams, and they're now encountering 2 sources of this same compound that they're attracted to. And they're doing this in 2 different contexts. First, when they're looking for tributaries that support larval habitat where their own offspring might thrive. And second, when they're looking for males. And I'm going to kind of talk about each of those contexts and think about a couple of things. First of all, what are the potential costs to females to responding the same way to this same compound, but from these 2 different sources? And then also all those costs are just presumed. We've never measured any of them, but kind of to support that, ask the question of whether they can discriminate between these two sources of the same compound. And when I answer those questions, or think about them at least in both of these contexts, starting first with the sexual context. So in this case, I would suggest that it's likely, again, that females benefit from this signal that brings them to males, but that it was probably confusing at first because they're encountering 3-kPZS from males, which they're looking for, but they're also encountering 3-kPZS from larvae, which at this point they're not looking for. And it's important to note that although the microhabitats that larvae reside in and that males construct nests in are very different. The larvae, as you can see in that picture, live in this really mucky habitat where the males construct their nests on these gravel bars. However, they're often interspersed very closely with each other. So in this is actually an illustration. Oh, you can't see it. Well, that figure on the right is an illustration of the stream that I do a lot of my work in, the Akiak River, and this was done in the early 50s when a pioneer lamprey biologist, Vernon Applegate, kind of did a lot of these basic studies on their biology. And this is just to show you, those are all spawning nests that males have constructed, and right downstream of them is where the larvae usually drift after they hatch, and they're very passive at this point. So just downstream of spawning grounds, there's usually larval habitat. But then even interspersed within those nests, there's often little patches of just mucky vegetated areas where the larvae really like to be. So it's very likely females are encountering 3-kPZS from larvae, which they're not interested in, while they're tracking 3-kPZS from males. So we then ask the question of whether females can discriminate between these two sources of this same compound. And we've done a little bit more than this, but just what I'm showing here is kind of the basic question of, do they respond to these odorants when we adjust so they have the same concentration of 3-kPZS? So this isn't a preference test. This is a no choice. Well, their other choice is just nothing. So I'm not comparing the responses, a preference between male and larvae. This is just natural odorant that we measured 3-kPZS in and then applied it at the same concentration. And as you can see, basically anything above 0 is a preference for that odorant. And as we've shown many times, they have a strong preference for male odor. But then, really interestingly, when we apply natural larval extract at the same concentration of this compound we know they really like, they show no response. And this is, in sea limpet, they're not like sophisticated like insects are, where we need like specific ratios and blends where it's off. They generally go to things they like regardless of what else is there. So this indicated to us that there's something else in larval odor that's essentially turning female responses off when they're looking for males. And we actually had a lead on what this might be because we had recently found some evidence that the precursor to 3-kPZS, PZS, acts as a behavioral antagonist to 3-kPZS, specifically interacting with some of the same receptors. So we thought PZS might be a mechanism where females could only respond to male odor rather than larvae, because we also had previous data that larvae release about 10 times as much PZS than 3-kPZS, where males release about 100 times more 3-kPZS, the attractive component, than PZS. And that's a tiny figure, but when we recreate those ratios for the larval ratio of 3-kPZS and PZS and the male ratio of 3-kPZS and PZS, we saw that 100% of the females that we tested in this in-stream behavioral assay went to the male ratio, even though the 3-kPZS concentration was the same in both nests. So it indicates that PZS is a mechanism that allows them to discriminate between these two sources of this deceptive but attractive compound. So the next thing I want to talk about is in the non-sexual context, when females are not looking for males, but rather they're looking for offspring that is a good place for for habitat that's good for rearing offspring. And in this case, I'd suggest that it's unlikely that it's costly for females to respond the same way to both sources of 3-kPZS. And this is for a couple of reasons. First of all, like I mentioned, during this non-sexual migratory period, they don't show a very strong targeted attraction to the source of 3-kPZS. So when they're prior to spawning, males signaling with 3K-PZS, they won't be bringing females right to their source. And even if they did, the biology of lamprey wouldn't allow them to coerce females into mating anyway. The second reason is although we think that, that migratory lamprey are attracted to the larval queue to find a good place to rear their offspring, the males releasing the same compound, it's still, those are both tied to reproduction. Whether they're looking for a good place to raise their young or a mate, it's likely that those kind of end in the same result, even if there might be different information associated with them. And I'd also note that males are also attracted to the larval queue. So females following males are probably following males that already followed the queue that they were more interested in the first place. So do we see support for this when we test whether females can discriminate? We did this in a larger scale test because, like I said, the migratory movements are a little more general and take place at a different scale. So we go out into the river and we have this, this site that we've used for decades now, and we can divide the stream into 2 channels and apply a different odor into each channel. And then we release migratory females at night and look at how many move up and enter each one of those channels. And this figure is just showing the proportion of females that enter each channel. And first of all, here I'm just showing that there was no strong bias for one channel over the other, although we did still alternate between them. So the next thing we did is we tested the larval ratio of 3K-PZS and PZS, the male ratio of 3K-PZS and PZS, and in each of those cases, it was just compared to nothing, so just to see if they responded to it. And then lastly, we applied each of those ratios in a different channel to see if they discriminated between them directly. And what we saw is that migratory females responded the same way to this mixture regardless of whether it was the larval or male ratio. And on the far right there, they actually didn't discriminate between it at all. So they aren't showing the same response that spawning females are. So to kind of put this together, I think we see that there's this indiscriminate attraction of females during the migration to the 2 sources of these compounds, the original non-sexual source of larvae and the novel sexual source of males that are signaling with it. But during spawning, females have specifically decoupled both sources of 3K-PZS and respond just in a way that's appropriate for mate choice. And with that, I want to acknowledge a bunch of co-authors on the papers that covered some of these data, and in particular, call out Skye Fissette, who was a graduate student that recently graduated out of the lab and really did a lot of this work, and then the funding from the Great Lakes Fishery Commission. And I will take any questions.
So we have time for one question or perhaps 2 short questions.
So there's other native lampreys. Do they respond to the same cue, or do
Yeah.
they use a different cue?
So the queue, they all respond to it. Like the larval queue seems very unspecialized. So it's like chemically we can measure the same compounds from different species, and we don't have a ton of behavior data, but what we do have shows that migratory females respond to those same cues regardless of what species. Yep.
Okay.
All right, thank you.
No, I have one. So could you design a compound that targets the same receptor so as to attract them with even more efficiency? So could you make a sensory trap?
Possibly. So I mean, we haven't tried that. A colleague in the lab, Ann Scott, is doing the opposite, where they're trying to design a compound that stops their response to 3K-PZS for management purposes. And that's the reason we have originally found that PZS does that, is because it seems to, but they're now doing some like chemical engineering to try to design better antagonists.
All right.
Yeah.
Good.
All right, now thanks.
Thank you.
All right, cool, perfect. I think we're right on time, too. Cool. All right, well, thank you all so much for coming. My lab is broadly interested in how genomic mechanisms generate and shape diversity, but I was super, super excited when Tino and Helen reached out and asked me to be part of this session because it allows me to go back and revisit some of the work I'd been doing before. I sort of have been transitioning in, and our lab is just starting to get back into visual ecology, so it's a perfect timing for that too. So at the beginning of this session, Becky was giving a really nice overview of sort of different models and ways that people think about sensory bias. And for the purposes of this talk, I wanted to do something kind of fun and weird as I'm looking back on some of the work that I'd done. And so I'm going to use a definition that's sort of the sensory bias model for the evolution of mate preferences posits that signalers manipulate receivers by exploiting pre-existing preferences for specific signal traits. And I've heard this one around in the field. I've heard other people use it. But there's one thing about this that has always kind of like irked me a little bit, and that's this part right here, that signalers manipulate receivers. And to me, it sort of sounds like, well, shouldn't selection now be acting on receivers to escape this manipulation? Anytime that we start to think about like a manipulation is that they're trying to get them to do something they don't otherwise want to do, which is why it was so great. I loved your talk just now. It was awesome. So I always got stuck on this, and yet what I keep thinking of is that we see mate preferences that are based on what we think of as these sensory biases observed in many, many systems, and they're persisting over very long periods of time. So somehow that's not all lining up. So what's sort of going on? Are there constraints to preventing receivers from escaping exploitation? And that's what I wanted to do, is look back at some of this work and try to ask, well, do we have any evidence that there could be some of the trying to escape this? And as I was thinking more about this, I started to think about how could receivers escape this manipulation? And I sort of came up with two alternative mechanisms that jumped out. The first being that they could change peripheral tuning. We're going to talk a lot about peripheral tuning. We've heard some about it already. And that's to say that as long as the signals from the signaler are not an exact match, then they should be able to retune their peripheral sensory system to distinguish between the ancestral bias and the trait that's trying to be used to exploit their system. And more specifically, I'm going to focus today on the visual system, so we'll get there in just a minute. The other thing that jumps out to me is we could be changing these higher order weightings, as how I like to call it, and that's to say that you can escape manipulation if you just change those sort of neural networks so you're not paying as much stock into this bias. You're essentially getting rid of it at a higher order. And I think this is sort of where the field is going. Part of the session was kind of a cool suggestion to put forward. Where is the field going to go in the next few years? And I think this is going to be a really cool and exciting thing as we're seeing more and more sort of circuits and neurobiology being brought on with non-model systems, traditional non-model systems. So I'm excited that it's coming, but we're not going to talk about it today. So for today, we're going to focus up here on how individuals can change their peripheral tuning. Specifically, we're going to focus on biases toward particular color, and then we're going to have to, of course, zoom in on color vision. And more specifically, we're going to talk about guppies, Basilia reticulata. These are small, live-bearing fish that I think a lot of people in this room are very familiar with. There's a very strong role of female mate choice that's going on. And thanks to work by Helen and others, they are one of like the best textbook examples for sort of a sensory bias for a particular visual trait. Okay, so being sort of a color-based signal, let's talk about how color vision could potentially be tuned in a way that we might look for ways that females are trying to escape a potential bias that males are taking advantage of. So we had a great, you can tell I came from Becky's lab. It's like the same figure, you know, here 15 years later. We had some great descriptions of how color vision works earlier in the session, but just so that we all remember, you have two cone cells. Those two cone cells will maximally detect light over different wavelengths. If we shined a 600 nanometer wavelength light at it, both of those cone cells might fire. They'll send that signal up to the brain, and your brain will say, yay, there's yellow. Cool. And once again, humans, of course, that have full color vision, we have our 3 different cone cell types. That lets us see our guppies here with these nice oranges and greens and things like that. But if we lose just one of those cone cell types, of course, we've lost out on those reds and those oranges, and even some of those greens. So that brings us to guppies, and so this is where we get to, do a one-up on the bluefin killifish, and, 5, but... so guppies have 9 cone-opsin genes. We generally have only 5 cone classes because they're not all expressed at the same time, okay. But it's really remarkable how many opsin genes they actually have. And one of the things that always has jumped out to me is that, yes, they go a little bit further into the ultraviolet. Yes, they go a little bit further into the longer wavelengths, but not extensively so. The vast majority of this duplications of opsins is actually sort of doubling down on that middle range. And so in theory, they could be using those to get a more fine discrimination between wavelengths of color cues. So what's one way that females could be altering their visual system to try to escape these biases? Well, one could be through differences to opsin gene expression. And so this is the idea that you might have, say, for example, 4 different opsin genes, but express only 2 of those. Having 2 totally different sets would be one way to do that. And to look at this in guppies in Trinidad, so this is the island of Trinidad. The Northern Range Mountains span along the top, and the watersheds will flow either north or south. The really great thing about Trinidad is that it's these independent evolutionary replicates where they've been independently colonized, and we can now ask for sort of experimental evolution that's naturally happened. Because in these watersheds, there are these waterfalls that separate these populations above the waterfalls having relatively low predation, and so they have low natural selection against colorful males, and males start to exhibit a lot of those red and orange colorations that we think that are tapping into this female bias, this sensory bias for the females. Meanwhile, below the waterfalls, we have high predation, and there's really strong natural selection acting to sort of decrease the male's red and orange coloration. So that brings us to a sort of, we can start to make some predictions about whether this escape is actually happening or not. And we can say if males are actually exploiting these females' sensory bias towards reds and orange, and if the females are trying to escape, if there's selection for them to escape, such exploitation, then we should see differences in opsin expression. We should expect also no differences in color preference outside of the mating context, because that wouldn't be changing the underlying bias. It would only be changing the discrimination for the males themselves. That there should be replicated differences in opsin expression between these high and low predation environments, and that females should have a weaker preference for red and orange males in the low predation populations. So any guppy biologist in here, of course, is like, but that's not what we see. We already know that. I also didn't measure that, so we've already sort of started violating our assumptions. So we might get to the end of this talk and be like, never mind, it's not there. So one of the things that we did as we were trying to explore this is we went to these populations across Trinidad, and we put our wonderful painted dimes a la Helen, and counted how many times did the population actually come by and start pecking at them. And we counted how many they would peck at each of the different colors. We then took a bunch of individuals to look for differences in opsin gene expression to ask if there's differences in tuning going on. As one that's worked with guppies before, wouldn't be surprised to know that guppies have a strong bias towards orange in all 14 of our populations. So what we're looking at here is populations along the X. On the Y is the proportion, or the mean number of times that colored penny was pecked at. And so all of them are orange bars. So if you're in the back and you can't see, they're all those big bars are pecks at the orange penny. And what was kind of surprising is that we didn't see a pattern between high and low predation populations for opsin expression across our 8 watersheds, which is something that we'd seen before when we used a smaller sample size. So we're still trying to understand how much is this a variance thing going on and how much is independent light environments changing things. But the other thing that we found was that opsin expression didn't correlate to color preference across the 14 populations, probably because we didn't have a lot of variance in our preferences for orange. So Helen and Mitch did a really awesome job of looking at this into the lab where we have a bit more controlled environment. And now we can actually measure the same individual for opsin expression and color preference. And so they very carefully would set up the pennies, give them the color preference, and then take eyes and send us the eyes for qPCR. And lo and behold, yes. So across individuals, we did see a positive correlation between preference for orange and expression of the long wave sensitive 1 opsin. So this is especially interesting because this is the furthest long wavelength opsin that the guppies have available. It's also the long wave sensitive that they express the highest overall. So it was like, cool, at least we're seeing something. The other really exciting thing was that individuals had, there was a positive correlation between preference for green and expression of the RH2 opsin, which is, as you might guess from the colors, tends to be maximally detecting light around that green wavelength. So cool, it's great. It says that, okay, opsin expression may actually mean something, which is good to know. But what does this mean back to our sort of sensory bias and escaping that? Well, maybe females could be altering their visual tuning through expression to mediate these biases, but these biases in opsin expression, changing, and tuning need to be explored a lot more. And one thing that this always brings back to me is, what does opsin expression really tell us about visual mechanism? We're often using, as a field, we're often using opsin expression as sort of this proxy for relative cone abundance in the retina, which makes some pretty big assumptions. One, that all the opsins are on the same diurnal rhythm. 2, that all cone cells express the same amount of opsin. And 3, that all cone cells express only a single opsin. And there's been, I think, 2 or 3 labs that have really actually looked at this in their systems. Becky's is one of them, so yay. But there really needs to be a lot more before we really dive into this, like using opsin expression to explain everything. We need to understand what we're actually measuring. Okay, so that's my side ramble. So back to the other ways that we could actually, females may be escaping this sensory bias. They could be changing their visual tuning through opsin gene sequence. And as you change gene sequence for the opsin genes, you're going to change the wavelength sensitivity of that opsin protein. And one of the questions that I had here that's always kind of tickled the back of my mind is, maybe the genomic architecture influences the female's ability to escape manipulation at sort of the species level. And this is because opsins, similar to olfactory receptors and most opsins, tend to occur in these long tandem arrays where we have gene copy, gene copy, gene copy. And one thing that tandem arrays tend to have a lot of is a process called gene conversion. And so if we look at just our long wave sensitives opsins in guppy, we see that we have LWS 1, 2, and 3, and especially when 2 genes are in a head-to-head orientation, what happens often is you have a random double strand break, and then when the template that goes to repair that double strand ends up being the wrong locus, you end up essentially overriding. So you're copying over what that copy was. So instead of divergence and having differences in opsin gene sequence, you've now homogenized all your opsins. So we asked if there is selection for females escaping being manipulated, then species with this red and orange males should show less LWS gene conversion so that they're maintaining a more broad opsin repertoire. And we looked at, I think we had 16 species here. And sure enough, it was actually really exciting to see that the species that males tend to have reds and oranges have very small regions that are undergoing gene conversion. And so that's what we're seeing here in red is how much of that gene is undergoing gene conversion. Cool. So does this framework of genomic architecture influencing females' ability to escape manipulation mean much beyond guppies? I think it does. Like I was saying, olfactory genes also tend to be in these tandem arrays. But maybe selection is going to be needed to continually purge these conversion events if you're actually in a species that's trying to escape this sort of homogenizing process. So real quick take-homes are, if sensory bias models are truly a conflict between signalers exploiting the biases of receivers, then maybe these receivers should evolve mechanisms to escape such exploitation, and biases should actually be lost. and also that it's possible that these proximate mechanisms of sensory bias constrain such escape, but these are largely still sort of black boxes. And opening these boxes is going to require some careful studies that couple measures of signal bias with measures of sensory system variation from the same individual or from the same populations at least. And with that, I'd like to thank collaborators and my lab.